warahmatullahi wabarakatuh and selamat sejahtera. I bid to Puan Marina binti Mota. We are from Group 2. In this video, we're gonna presenting about issues in teaching and learning in school, chapter Cell Division. Members in team. We have Raihana, we have Mega, we have me Zadiatul, and we have Vaishnavi. In this video, what are we going to learn? The objective is to have better understanding with more precise on these following 17 terms.
So now, I'm gonna explaining about chromatid, chromosome, centromere, sister chromatids, chromatin, homologous chromosome, and tetrad chromosomes. In all eukaryotic cell, we have a nucleus in our cell, and in that nucleus, we have chromosome. A chromosome is a long DNA molecule with part or all of the genetic material of organisms. The centromere are located at the middle of the chromosome. As you can see, the chromosome on the left side is before replication and the right side is after replication, after the chromatid replicate itself into two. A chromatid is one or two identical halves of a replicated chromosome. As you can see in the picture, before the replication, one chromatid per chromosome, and after replication, it has two chromatid per chromosomes. And the two chromatids are called sister chromatid. Chromatin is a substance within a chromosome consisting of DNA and protein, which are called histone. So, chromatin is actually a semi-condensed form made of DNA plus proteins. Homologous chromosomes are two pieces of DNA within a diploid organism which carry the same gene, one from a parental source. In simpler terms, both of your parents provide a complete genome. Tetrad chromosomes. When the homologous chromosome undergo crossing over in the metaphase 1 stage, it will be tetrad chromosomes. Tetra means 4, so it has 4 chromatids that are aligned together. For example, at the first chromosome, it has 2 chromatids, the blue and the red. And at the second chromosome, it has 2 chromatids too, the red and the blue. So, total we have 4 chromatids in the tetrad chromosomes.
Haploid cells. Haploid cells are cells that contain only one set of chromosomes and are formed by the process of meiosis. In higher organisms such as humans, haploid cells are only used for sex cells. Examples of haploid cells are gametes. Diploid cells. Diploid, as the name indicates, it contains 2 sets of chromosomes. Diploid cells undergoes mitosis. In higher organisms such as humans, all other cells besides sex cells are diploid. Example of diploid cells are blood cells, skin cells, muscle cells, which are also known as somatic cells. Gamete cells. Gametes are reproductive cells such as sperm and oocyte, or better known as the egg. Gametes are haploid, which means there is one copy of each chromosome in each gamete. This is important because humans are going to reproduce to form a diploid organism. Somatic cells are cells of the body. In humans, somatic cells are diploid, which means they contain 2 copies of each chromosome. Human somatic cells have 23 pairs of chromosomes for a total of 46 chromosomes.
kinetochore microtubules who are positive and are attached to sister chromatid pairs of kinetochores. Kinetochore has microtubule attachment and control the movement of chromosomes during mitosis and meiosis. An animal cell kinetochore has 10 to 40 attachment sites. The attachment site has a protein collar that secures the microtubules. Kinetochores allow kinetochore microtubules to attach correctly to sister chromatids. If there's incorrect in microtubules attachment, such as the microtubule from the opposite spindle poles attached to the same sister chromatids, the attachment is unstable, and it's the other way opposite where the attachment is stable. Spindle microtubule fibers grow out from the centromere and all polymers composed to alpha and beta tubulin subunits. A microtubule has distinct polarity. The growing end is termed the plus end and the minus end associated with the centromere.
We're going to talk about the term cytokinesis. Cytokinesis is a process where the cell will divide into two. Where the two nuclei are already on the opposite pole, the cytoplasm separates. The cell will pinches in the middle, leading to cleavage. So, what is cleavage? Cleavage occurs by the contraction of a thin ring of actin filaments that form the contractile ring. The ring contracts and eventually pinches the cell until it separates into two independent daughter cells. For example, a symmetrical cytokinesis can be formed in the process of spermatogenesis, where the newly formed sperm cells are equal in size and content. Next is the karyokinesis. So karyo means nucleus and kinesis means division. So basically it's a division of nucleus, also known as the indirect nucleus division. The nucleus from the parental nucleus will divide into two daughter nuclei. It is a continuous process without any pauses in between the whole process. It has been subdivided into four major phases, which is prophase, metaphase, anaphase, and telophase. Now we continue with chiasmata. Chiasmata is the point of contact, the physical link between two chromatids belonging to homologous chromosomes. At a given chiasma, the exchange of genetic material can occur between both chromatids. In humans, there seems to be one chiasma per chromosomes, meaning that the DNA is exposed and exchanged only that site a minimum at once and rarely more than 3 times. Through this, we can see the importance of chiasmata. Now we continue with crossing over. So crossing over is the process which produces recombination of genes by interchanging the corresponding segments between non-sister chromatids of homologous chromosomes. So a crossing over between linked genes allows their recombination during meiosis.